thank you to the CTNNB1 Connect and Cure community and the Med13L Foundation. This will be really clear in the next couple slides, but this project that I'm going to talk about quite literally could not have been done without both of your foundations recruiting and, more importantly for me, trusting us with your kids and your families. And so basically everything I'm about to show you is really made possible by these two foundations as well as the others who are participating in this project. So as Kellen already introduced for me, as well as Rachel, is what I'm going to talk to you today is just to show a little bit about the cross-disorder biomarker work that we've done at COMBINEDBrain, leveraging changes in plasma proteins as the tool to identify candidate biomarkers for rare diseases. So Kellen just showed this slide, but it is really my favorite slide to show an overview of the drug development pipeline for rare genetic diseases in particular. Okay, I'll pretend like I have a pointer. On the left-hand side of, oh wait, maybe I can use this? No. On the left-hand side of the screen, you can see where you identify the pathogenic gene that's causing the particular symptomology. And then as you move to the right, you conduct various basic and clinical research efforts that get you towards clinical trials. And then, as Kellen already pointed out, one of those really critical steps is identifying biomarkers for that particular disorder. So I know I don't need to go into this with a lot of detail with this particular group, but biomarkers are generally thought of as objective measures which can be used to predict disease status, so disease severity, disease progression, as well as whether an individual will respond to a particular treatment or not. And in the context of neurological disorders, we've seen these biomarkers be anything from different mRNAs or genes, microRNAs. We see various epigenetic modifications specific to disorders, proteins, metabolites, and even gut microbiome profile. So these are typically found in various biologic fluids. For neurological disorders, obviously, cerebrospinal fluid is a great source to find biomarkers. Blood, urine, saliva, stool as well. But one of the challenges for finding biomarkers for neurological disorders is to try and find a less invasive fluid or tissue type that can recapitulate what you see in the brain. And there are several examples of this in the literature. In fact, here are two figures that I pulled in another neurological disorder called Huntington's disease. But you can see on the slide, the figure on the right, I really wish I had a pointer here. Hold on. Okay. The figure on the right shows that there's a significant positive correlation between cerebrospinal fluid, neurofilament protein levels, and plasma protein levels. And then the second one shows that those plasma levels actually correlate really nicely with indicators of clinical severity. So we know that there are things that change in the plasma that can help predict sort of disease severity and status. And this is really useful when we're starting to think about what to measure in clinical trials to determine efficacy. So one of the biggest hurdles to finding biomarkers for rare diseases is, as the name sort of suggests, is that there's really limited access to patient samples when you have a rare disease population. So COMBINEDBrain established a biorepository. And now I think I've utterly wonked my slides here. Sorry, Rachel.
Oh, it got disconnected.
I broke it.
She broke it. Yeah. Oh, wait. Nope. I figured it out. I just read. We're good. Maybe. Yeah. It's not picking it up.
I think Rachel broke it now.
We have a few minutes, so.
Yeah, so I have a repertoire of knock-knock jokes I could go to.
Yes, all of them.
Why don't we start over? From current slide, maybe?
Yeah, right.
And then what
Yeah.
happened? Now it's moving.
No, not up on the screen. It's frozen.
Resume slideshow.
Nope. Geez, Anna.
Whoops.
Thanks for coming, everybody.
Okay, I don't know why that
Pay
happened.
no attention to my desktop. It's a mess. Oh, you know what? That's an extender, I think. That's a,
Well, right, we're mirroring it,
Yeah.
so we should be able to put it in. Is it working? Good.
yeah. And then here's your laser pointer, Anna. And you can, so you should just be able to, get your pointer up and then,
Oh, okay. And then do I press forward?
yeah.
Okay.
Yeah.
Okay, yeah, that's fun. Great. Way to nail it, second thing of the day. Okay, all right. So everyone remembers what I just said. So we've been collecting patient samples from rare diseases for the last 2 years and really focused specifically on designing a project that might help us start identifying biomarkers for rare diseases. And so what we did for the project that I'm going to talk about today is we set out a goal to collect 20 unique patient samples from as many rare neurological disorders as possible, as well as 40 neurotypical, typically these are siblings, but aged and sex-matched, unaffected individuals. We didn't have any age or sex matched restrictions on this, so we collected individuals anywhere from 0 to 60 years of age. But for the data that I'm going to show today, since I was really interested in looking at changes that happen during neurodevelopment, we only are going to show data from the analyses that we've done from individuals who are between 0 to 17 years of age. So we collected a blood sample, processed it into plasma, and used a small portion of that plasma to run proteomics on this SomaLogic platform, which allows you to measure about 10,000 different analytes measurable in human plasma. And in addition to the plasma sample, we also requested caregivers to complete everybody's favorite neurological assessment, which is the Vineland. We had caregivers complete the Vineland, which is canonically a measure of adaptive behavior, but has been used historically in the context of neurodevelopmental disorders as a readout for neurodevelopmental status. And then we also asked individuals to complete the PELS seizure survey if they reported epilepsy in their child. So for this project, I had really essentially 4 basic research questions, which was, do we see similar changes in plasma proteins across all of our affected disorders compared to controls? Do we see some unique proteins that change by distinct disorder? And then how do plasma proteins or plasma protein profile change with age, and how does epilepsy change that? So the way that we collected these samples is a variety of different ways. The vast majority of those samples were collected by us flying a research team to 19 different conferences. You can see the states colored here. And we collected blood on site and processed it. We also were really diligent about trying to make sure that we captured individuals who were very severely impaired. So we actually sent mobile phlebotomists to patients' homes to collect this type of sample as well. And then we also collaborated with a variety of medical institutions to piggyback off of clinical labs that were already being drawn. So in the span of about 10 months, we actually collected a little bit over 700 samples. And the table that you're looking at right now is actually the breakdown in enrollment for individuals between 0 and 17. So the top bar shows the number of control samples that we collected. And then as you look down this table, you can see the 28 different neurodevelopmental disorders that participated in this project. I've highlighted CTNNB1 related disorder samples as well as MED13L related disorder samples. There were 26 CTNNB1 donors in this age range and 18 MED13L related disorder samples. The age was almost exactly the same, a little bit younger than 7.5. Almost approximately the same age breakdown. About 4% of individuals with CTNNB1 reported epilepsy compared to about a quarter of MED13L kids. And then these three rows on or columns on the right represent the different composite domains of the Vineland: the adaptive behavior composite, communication composite, and daily living skills composite. And I'll go into it a little bit more detail on the next slide, but essentially, as a general rule of thumb, the lower your score, the more cognitively impaired you are. And a general rule is that if you score less than 70, you're considered cognitively impaired. So here is a figure that shows all affected samples and how they score on this adaptive behavior composite. The threshold of 70 is highlighted in blue, and you can see that people are significantly lower than this line. And we also were really interested in looking at communication as well, because that is a symptom that's most burdensome and reported to families from a bunch of patient surveys that we've completed. And so you can also tell that all of the affected individuals scored significantly lower than what you would experience from neurotypical individuals. The other thing that we wanted to look at was what specific parts of communication might be impaired. So if you look at this dotted gray line right here, this shows the rate at which neurotypical individuals would gain receptive communication skills, and the same thing with expressive communication skills. And you can see that all the colors at the bottom represent what that slope looks like with various disorders. And I broke it down for MED13L and CTNNB1 related disorder on the far right figures here. And what you can see is that the rate at which they gain communication skills is significantly lower or slower than neurotypical individuals, but they do gain skills with age. Okay, so I actually have like 4 more slides, but I'll go fast. So one of the main research questions was, is the plasma proteome similar amongst disorders? And in order to look at that, I wanted to really see, are there major differences in sex, age, and epilepsy? And so what you see here, these are UMaps, which are a way to sort of visualize the variability or the groupings in your data in a 2-dimensional space. And you can see that sex, between males and females, red and blue, there's no significant differences or clusterings by sex. There's also no significant clusterings by age, so different colors of blue show increasing age. And then very interestingly, there's no clear pattern or impact of the plasma proteome on individuals with epilepsy. So individuals that are sort of white marks don't have epilepsy. These gold circles here represent individuals who have epilepsy but haven't had a seizure in the last 60 days. And then red individuals are people who have had some form of seizure in the last 60 days. So one of the most interesting findings, so this is the same UMap, although in this case, the different colors represent different individuals with different disorders. So each disorder is a different color. And without going into any sort of detail, what you can see is that there's no groups by color, which would suggest that there's no clear distinction in plasma protein profile by genetic etiology. So because we saw that, we thought, okay, let's put all of our affected samples together, compare them to age-matched controls, and see what proteins are upregulated or downregulated. And what you get when you do that is you get that there's 160 upregulated proteins and 72 downregulated proteins, which is quite a bit, and we're still filtering through what some of that means. But what you can see is some things that I've recognized in the context of a neurological disorder have been altered in the past in plasma. And those are these SAA1, serum amyloid alpha 1 and 2, as well as glial fibrillary acidic protein, GFAP. So just for the sake of time, I won't go into great detail, but the disorders are listed very smallly here, which I don't expect you to be able to read. But the disorders that have this red highlight right here show that, in fact, these levels are significantly altered in the plasma of these patients. And among those groups that are highlighted, if you can't see it, is CTNNB1 and MED13L. Okay, so the next thing that I thought was, okay, if samples don't separate by genetic cause, maybe they sort of group together by shared molecular function in the sense that what the pathogenic protein does in a normal context in the cell. So we separated the groups that were participating in this project into 8 different sort of molecular categories: transcription factors, channelopathies, chromatin modifiers, copy number variants, synaptopathies, transporters, kinases, and disorders that impact myelination. So I'll focus on the transcription factor group just for the next 2 slides, which are my last 2 slides, because MED13L and CTNNB1 are both, in this context, considered transcriptional regulators. What you can see on the same colorful figure that I've been using several times over is that individuals that have CTNNB1 related disorder, which are these orange dots, cluster quite distinctly, in this case, from individuals who have MED13L related disorder. So this would be a sign that perhaps they are not as similar as you would expect. And in fact, CTNNB1 clusters much closely to something called WOX-related epileptic encephalopathy, and MED13L actually clusters closely to HNR and Ph.D. related disorder, which Jen knows a lot about. Okay, so this is one of my last slides. So the very, very last thing that I'll show today was just looking at what changes on a by disorder basis. So what disorders change in CTNNB1 related disorder compared to age matched controls. And what you can see is that there are a handful upregulated, more downregulated proteins. And the 2 proteins that I pulled out, not necessarily because they have funny names, but they're from the Homer family, is because Homer proteins are almost exclusively expressed in the postsynaptic density of excitatory neurons. So we're particularly exciting in the context of neurological disorders. And so when you look at Homer expression with age, you don't really see any clear patterns with age, but you do see the reduction compared to neurotypical controls, which are the empty circles. But you wouldn't necessarily expect a change in protein with age for this particular protein. However, when you look at expression of this particular protein over the communication composite scale, now there's only 10 individuals in this case who completed the Vineland, but you start seeing some trends that might suggest that protein expression correlates with some indicator of clinical severity. Okay, so I'll go very fast for this last one. So I did the same exact thing for MED13L related disorder. And the protein that I highlighted here is this FZD10, which is frizzled, which Kellen already introduced and is part of the Wnt signaling pathway. Frizzled levels don't really change with age, but there is this really interesting, very preliminary correlation with FZD10 levels and communication composite, a marker of clinical severity in terms of communication skills. Okay, so very quickly, I call these current conclusions or next steps. We've identified several proteins which are enriched across a variety of different disease groups in our project. The hard part now is trying to figure out what's the most appropriate phenotypic measure to use for clinical severity. We are collecting cross-sectional samples. We need additional cross-sectional samples, but probably also, more importantly, need longitudinal samples. And then the next part of this project is to try and validate some of our findings in the plasma in cerebrospinal fluid samples. And then I think one of the most exciting things, as Kellen alluded to, is that there is preliminary data, that there is this sort of overlap and pathology between MED13L and CTNNB1 in terms of implicated Wnt signaling disruption. Okay, I know I'm over, but these are the people who actually did the work, and so I'm going to thank them. I have an incredible group led by Sasha, Grace, Danielle, and Taylor, and here's Rachel dressed as Mario, so you're welcome. Rachel. Thank you to Simons, who allowed us to share some of our phenotypic data and shared Vinelands from completed participants. Jason for setting up our online data collection platform, our computational team of gurus, Ricardo, Megan, and Daryl, without whom I would have 0 figures to show you today, and then Natasha for her sort of very quick, awesome crash course on understanding how to interpret Vinelands. My funding partners, and then obviously all of the foundations that participated in the project. Okay, done.